Now I will hand in the chair to you for the next speakers.
Sure, certainly. All right. And it gives me great pleasure to highlight the other speakers in this session. Let me just pull out. I think Phil will be going next. Phil hails from the Queensland University, and his topic will be preservation of fecal microbiome for metagenomic analysis, which I think flows very nicely from what I had shared. I have highlighted all the problems, and now Phil will share with us what the right solutions are.
No, maybe. Can you hear me?
Yes, we can.
Okay, and what are you seeing currently? Are you seeing the main presentation display, or?
Yes, we are seeing it in full screen mode. So everything.
Great, thank you. Great. Yes, so I agree. Jeremy's talk and I will overlap very nicely, I think. So today I'm going to talk about looking at some different preservation methods for the fecal microbiome. My day job, I'm a professor of microbiology at the University of Queensland, but today I'm speaking in the capacity of the company Microba, and Microba is a gut metagenomics company that I co-founded with Gene Tyson, so that's the disclaimer. And really what I want to do is draw your attention to a study that you may have missed, judging by the number of citations. It looks like a lot of people missed it, but we did as part of the company work, we did a critical evaluation of fecal microbiome preservation methods specifically for metagenomics. And the work I'm presenting today is mainly Eleanor Pribble. Many of the slides are hers, but this whole team contributed to put this story together. And I'm only telling you part of the story because it's 15 minutes. So I would direct you to the paper to get the more complete story. So I don't need to tell this crowd that the number of gut microbiome research articles is really increasing at a high rate of knots. This is already 3 years out of date, but I imagine that the curve is continuing straight up. And many studies, as Jeremy just pointed out, rely on remote collection of fecal samples. And from the perspective of the company, we're interested because we send out a kit that people then mail back their samples through the mail. And so we really wanted something that was simple, didn't need freezing, that could be done at room temperature or higher because it's in Australia. So actually the ambient temperature, particularly during summer, for collecting samples could get very high. And I'll talk briefly about that towards the end of the talk. And many studies assume that sample collection methods not distorting the community. But as Jeremy pointed out, I think there is quite a strong realization that this is a very important and basic feature of these type of analyses that has to be taken into account. So there are multiple methods for room temperature sample collection, which we were specifically interested in. And Jeremy's pointed out a few of these already. So it's important because if these skew the community, it can create false leads and you can miss key biomarkers for the biological question you're asking. Surprisingly, there have actually been very few studies that have assessed room temperature preservation of fecal samples specifically for shotgun metagenomics. Most of the work has been done with 16S, which I guess is understandable because that's still dominant in the field. But we're interested in metagenomics because metagenomics also tells us about, say, functional profiles in addition to who's there. And there was actually only one study using metagenomics that assessed technical reproducibility of this collection. So this is the study I just highlighted before. So what we did was we looked at 5 room temperature methods in order to see which is the best performing one for fecal sample collection. So this is the study design. We had 5 healthy individuals provide a whole stool sample, which was homogenized. And then we took 6 replicates across 5 different methods. So 3 of these are dry preservation methods, no liquids involved. That's the ones we're more interested in. We wanted to reduce the complexity of the collection and not have liquids involved, but also 3 liquid preservation methods. So of these, Jeremy mentioned OmniGene gut, which is specifically for gut samples, RNA later. LifeGuard we also included. LifeGuard is typically used for soil samples and not for gut samples, but we included it because it's a well-known way of preserving samples for biological analysis. In the dry preservation, we just used the straight BBL swabs that's been used by the American Gut Project, for instance. Flash freezing, which is kind of the industry held up as the gold way of doing things. Obviously, not really feasible in the field. And I have something a little more to say about that. And then another technique which hasn't really been used for fecal samples, which is the flock swab ADT. That's actually a dry swab with a desiccation step for preserving samples. It wasn't designed for fecal samples. It was designed for forensics and taking samples from the field for blood splashes and stuff like that. We were looking to have the samples collected and then stored at room temperature for 4 weeks. Now, some of these kits recommend not to store beyond 1 week, so we did that. And for the ones that said 1 week max, we then stored it for the rest of the time at -20. But for, say, the flock swab, we could do the full 4 weeks. The OmniGene gut we could do for the full 4 weeks. And we did 6 replicates per method. Just a quick shout out, we did for the bioinformatic analysis of the data. So we sequenced 7 million reads, a minimum of 7 million reads per sample. That's about 6 gigabases of data per sample. Then we used a community profiler that's been developed by Donovan Parks at Microba. It's a commercial one, so it's not for public use, it's for commercial use. But we did publish a comparative analysis of different profiling tools for metagenomic data, and this was published in 2021 in Frontiers in Microbiology. So I just refer you to that. It's quite useful for looking at all the publicly accessible ones and looking at the strengths and weaknesses of the different profilers. But anyway, this is what we used, and I can vouch that it's a good bioinformatic workflow for analyzing these type of data. So cutting to the chase for a short talk, these are plots showing the Bray-Curtis similarities, and so the better the technical reproducibility, the lower these bars will be on the axis. So we were assessing them both in terms of the species profile and functional profile. So the genes plus at the species level. And the flock swab and the OmniGene gut had the best technical reproducibility, and the worst was the dry swab, which was somewhat anticipated. And then LifeGuard was the second worst there. I always, seeing is believing in my book, I like to see the actual profiles. So these are the profiles of the 5 individuals, and as is well known, actually most of the differences that we saw were between individuals. But within the technical replication, what we noticed immediately was that there were, the reason that the technical reproducibility was so poor on the swab, the dry swab, and in LifeGuard, which was surprising, was due to these sporadic outgrowths of facultative anaerobes in the samples. And so this is just a blow up of this, and you can see the 3 that we found in E. coli, well-known facultative anaerobe in the gut environment, but also another Escherichia species and Citrobacter freundii. And so we kind of expected this for the BBL swab, and in fact, the American Gut Project even produced a bioinformatic workaround to overgrowth of facultative anaerobes in those samples. But we were a bit surprised to see that these organisms can actually grow in LifeGuard. So that's a warning for anybody using LifeGuard that you can get microbial growth within the samples itself. So having established that what the best performance, we wanted to then go and check out what the range of storage temperatures for the flock swab might be. Again, because the idea that if we're sending samples back through the post in Australia, it can get up to 50 degrees when the sample is return sample sent in the mail. So we checked that, and in this case we took 10 fecal samples from healthy donors, and we did 6 replicates again, and we did fresh sample, a frozen sample, room temperature sample, and 50-degree sample all at 4 weeks. And so a total of 240 metagenomes in all. And cutting to the chase again, there was good technical reproducibility across all temperature treatments for both the species and the functional profiles. And again, seeing is believing, these are the profiles here. Very stable species profiles, of course, across all temperature treatments, even at 50 degrees for 4 weeks. So we're happy with that. Now, this is an interesting thing that came out of it. Freezing is typically seen as the best practice, but we did notice that there was consistent differences observed between the frozen controls and then our best performing in terms of technical reproducibility, the flock swab and the OmniGene gut and the RNA later. And what we saw that there were consistently elevated relative abundance of Prevotella copperi and Bacteroides species relative to the frozen one. And so, and there was almost no difference when the flock swab was compared to the fresh sample. So what we think is actually the freezing is causing a bias in the community profiles, because presumably because as the samples are stored, those particular type of cells may be particularly prone to more rapid lysis. And so when you thought to do your actual analysis, you're somehow along the way losing DNA from these, or identifiable DNA from these species. So the few studies have directly compared fresh versus frozen samples, but the ones that we did see, there was some trends for decreased Bacteroides and Prevotella. So that's just a cautionary tale that maybe freezing is not the best way of dealing with the samples anyway. And in fact, we can think of fresh samples as the best, but you can then use these other methods for storage for sending the samples. So just the conclusions. So we strongly recommend against using dry swabs, the BBL swabs and lifeguard, because they get unpredictable outgrowth of the facultative anaerobic species. We found the flock swab ADT was the best performing method, although the other ones, some of the other ones were quite good too. But the benefit of the flock swab, it's a dry method, so no liquid involved. So when you have customers or patients, it's a very, very simple approach just to have a dry swab to swab a piece of toilet paper. And we were very happy to see that the flock swab had high technical reproducibility, even if you had the sample stored at 50 degrees for a month. So we believe, in short, and we use this at the company, that the flock swab ADT, even though it wasn't designed initially for fecal samples, is actually an excellent option for remote collection of fecal samples. No liquids, easy to use, and can be stored at a wide range of temperatures for up to 4 weeks, and possibly longer. We didn't test it. Okay, that's it. Hopefully I did that well within 15 minutes to allow time for everybody. And I refer you to Eleanor as the contact. Here's her contact details, and she can tell you in exhausting detail about the statistics in the study and any other thing that you might be interested in based on the study. Thank you for your time, and I'm happy to take questions in the Q&A session too.
Thank you so much, Phil. Very, very illuminating, and I'm really glad that you took the time to walk everyone through the paper that you had shared, because clearly something that's easy to collect that is room temperature based offers much more potential